Welcome to the next session on biology, chapter cell cycle and cell division. The last module we had learned about the significance of mitosis, the steps of mitosis. In this module, we are going to learn about the next type of cell division that is called as meiosis. So where does meiosis takes place? It takes place in the germ cells. Germ cells means those cells which are involved in the formation of gametes. So it is taking place in germ cells who are in diploid condition. Diploid means twin, who's having two sets of chromosome, one from the parent, father, and one from the maternal parent. So paternal and maternal parent contributes equal set of chromosome, and thus they have two sets of chromosome. So this in this type of division, the chromosome number is half. It is reduced to half in the progeny cell. So if the parental cell is having 46 chromosome, then the daughter cell will be having 23 chromosome. So the number of chromosome is reduced to half. That means the parental cell is diploid in nature, whereas the progeny cell or daughter cell is haploid in nature. It's having only one set of chromosome. So this haploid daughter cells is nothing but the gametes. So such type of division in which the chromosome number is reduced to half is called as reductional division. And it occurs during gametogenesis for the formation of gametes in which the germ cells divide by meiosis to give rise to the gametes. It leads to haploid phase in the life cycle of sexually reproducing organisms. That means only the gametes constitute the haploid cells in our body, whereas all the other somatic cells and all the other germ cells in our body except the gametes are diploid in nature. So it is these cells or products of meiosis which represents the haploid phase in sexually reproducing organism. And after fertilization, which is fusion of the male and female gamete, the diploidy condition is restored. So the diploid cells undergo meiosis to form haploid gametes. The haploid gametes, they join at the time of fertilization to restore the diploid condition. What are the key features of meiosis? It involves two cycles, meiosis 1 and meiosis 2. So meiosis 1 and meiosis 2. But what we have to remember is the DNA gets doubled, or the replication of DNA takes place only once. Even though there are two cycles, meiosis 1 and meiosis 2, the replication of DNA takes place only once. And here there is a particular event called as pairing of homologous chromosomes, or it is called as synapses. And another step is recombination or crossing over occurs. So meiosis 1 begins after replication of parental chromosomes to form identical daughter chromatids at the S phase. So we know during interphase at the S phase, the DNA gets replicated to form identical daughter chromatids. And that is the only DNA replication which is taking place one, even though there are two cycles. And at the end of meiosis 2, 4 haploid daughter cells are formed at the end. So the salient features are two cycles are involved, meiosis 1 and 2, but DNA replication occurs only once. Pairing of similar chromosomes or homologous chromosomes takes place. Crossing over a recombination takes place. And meiosis 1 is followed after the replication of DNA in the S phase, and 4 haploid daughter cells or gametes are formed at the end of meiosis 2. So meiosis 1 occurs the same stages: prophase, metaphase, anaphase, and telophase. But what is the difference between meiosis 1 and 2 is that it is denoted as prophase 1, metaphase 1, anaphase 1, and telophase 1. Whereas meiosis 2 is represented as prophase 2, metaphase 2, anaphase 2, and telophase